Hello, everyone. We're going to get right into meiosis now. We just finished with mitosis. Mitosis took one cell. Hello, there we go. And produced two identical cells from that one cell that we started out with. Meiosis is a reproduction or a replication that takes one cell and ends with haploid, so depending on the type of meiosis, 1 to 4 cells that are non-identical. And in doing that, it reduces the number of chromosomes, which is what it says up here. We're reducing chromosomes. So this occurs in the life cycle of sexually reproducing organisms. And the reason we do this is to provide offspring with different combination of traits. So mom and dad gave genes to our offspring, and those genes are going to get mixed up, and the offspring will have unique characteristics that neither mom nor dad had. So we start with a diploid cell. So this diploid cell, I'm going to draw it over here. I'm using a red just because that's the color it was on. And I'm going to change the color to this blue. So diploid cell has a chromosome of each type from mom and a chromosome of each type from dad. And then it ends up with 4 haploid. So I'm going to draw those down here. I'm actually going to change the color of the ink. We'll go 1, 2, 3, 4. And each of those cells is going to have half the DNA. So there's that one, and I'll do that one again, and I'll do this one here, this one there, and then we'll do this one, that one, this one, and that one. So you end up having 4 non-identical haploid sex cells or daughter cells. Each of these has half the amount of DNA, has one of each of the homologous chromosomes. So remember, homologs are a pair of chromosomes, one from mom, one from dad. That's one set of chromosomes. Here's another set of chromosomes, okay? And so that's what meiosis does. We take one single cell that is diploid and produce haploid sex cells from it. In general, we produce 4 haploid sex cells. So meiosis goes through 2 sets of division. So we go through that interphase just like normal, and so we double the chromosome number, and all the chromosomes that are in the cell are going to be attached to their sister chromatid at the centromere positioning. Then we go through prophase 1. So during prophase 1, nuclear membrane breaks down just like we did in mitosis. Chromosomes are going to form, but in this case, the homologs line up next to each other and a process known as crossing over occurs. This is unique to meiosis. It does not occur in mitosis, and it only occurs in prophase 1. So we'll talk about crossing over in a little bit. But what crossing over does is it shares genetic information from dad and mom's chromosomes with each other. Then we go through metaphase 1. So in this case, in mitosis at metaphase, the chromosomes lined up one on top of another. So you had one chromosome, and then you had one chromosome and one chromosome lining up like that at that metaphyseal plate. In meiosis, they're going to line up like this. So I'm going to draw a line here. So you're still going to get mom's chromosome. And I'm just going to do this because I already have it there. So they line up together. And so here is that metaphyseal line. And the chromosomes are lined up next to each other, okay? So they line up together at that plate. So that's a difference that we see. And they can line up in any way they want. You can have, I put the red and the red on one side and the blue on the other side. It could have been red and blue. You could have had, blue and red. So it doesn't matter. They line up however they line up. And that's actually known as independent assortment, which again, we'll talk about in a little bit. So here you can see prophase 1. So we have this initial cell, and I'm just going to draw that initial cell over here after replication of DNA occurs, okay? So we have big and small in this case. So big is going to look like this, and small is going to look like this, okay? Just because that's how I made it. Oh, well, actually, I'm going to change it because you see the small are the same shape. So small will look like this. Okay, and then we have big blue and small blue, okay? And so this is right at the end of, or this is interphase. And the phase when the replication occurs would be during the S phase. Remember that, okay? So from here, we're now going to go through prophase 1. And in prophase 1, homologs come together. So the nuclear envelope dissolves. The centromeres with the centrioles are moving to the opposite poles. And the centrosomes with the centrioles move to the opposite poles, and the chromosomes undergo a process known as crossing over. Okay, so when this happens, we're exchanging information. I'll show you that a little bit later again. So this only occurs during prophase 1. Only occurs in prophase 1. Then we go through metaphase 1, and they line up at that metaphyseal plate, but the homologs line up together at the metaphyseal plate. And we now have the chromosomes are attached. And you can see that here. You have this set. So instead of having each sister chromatid attached, we have one full chromosome with both sister chromatids attached here, and then the other are attached here and here. And so then we'll go through anaphase, and in anaphase, homologs are separated. So each homologue still consists of its two sister chromatids, but they're separated from their other homologue. So instead of having the chromatids ripped apart, we have the homologues pulled apart. And then in telophase, the nuclear envelope can reform around, and cytokinesis can occur. And so this is going to produce two daughter cells. So here you see anaphase, you have cytokinesis starting, and here you have that new daughter cell, or the new daughter cells being formed, new nuclear envelope, but each daughter cell now is going to only consist of 1/2 of the amount of DNA. So you have big blue and little red, and big red and little blue over here, okay? And so now each of these cells, cell 1 over here and cell 2 over here, has to go through another meiotic division. So we went through meiosis 1, and then we go through a process, a rest phase basically, called intrakinesis. So during intrakinesis, the cells are going to get prepared to go through their second meiotic division, but there's no DNA replication that's going to occur. There's no crossing over that occurs because now we don't have the same information. They're different chromosomes. We only have half the amount of DNA. So we now have haploid cells that have duplicated DNA. And so now we're going to go through another meiotic division, meiosis 2, to separate the sister chromatids from each other. So this is very similar to mitosis. In prophase 2, the chromosomes condense if they're not still condensed. Nuclear envelope will break down again, and the centrosomes with the centrioles will move to the opposite poles. New spindle fibers will form. During metaphase, the chromosomes line up one on top of each other, just like in metaphase of mitosis, we're lining up one on top of each other in metaphase 2. In anaphase 2, sister chromatids are pulled apart. And in telophase 2, the new nuclear membrane reforms, cytokinesis occurs, and we end up having, so here's one of those cells, we end up having two non-identical daughter cells from each of those divisions, but you're going to have that other cell right here going through that same process. So remember, there was two cells at the end of meiosis 1, each of those cells goes through it. So we end up with 4 daughter cells that are non-identical and haploid. And so this is the entire process of meiosis. You start with this diploid cell. That cell goes through DNA replication, and then we have some unique activities occurring like synapsis and crossing over. And then we have a special lining up of chromosomes, which is known as independent assortment at the metaphyseal plate of meiosis 1. And then we go through meiosis 2, and we end up with haploid non-identical sex cells right here from the one single parent. So we have one parent cell that's diploid, goes through meiosis, and it produces 4 non-identical haploid sex cells. I'm going to stop here, and we'll get into the next video shortly. Bye.